Evolution of the wolf. The evolution of the wolf occurred over a geologic timescale of at least 300,000 years. The gray wolf Canis lupus is a highly adaptable species that is able to exist in a range of environments and which possesses a wide distribution across the Holarctic. Studies of modern gray wolves have identified distinct subpopulations that live in close proximity to each other. This variation in subpopulations is closely linked to differences in habitat, precipitation, temperature, vegetation, and prey specialization, which affect cranio-dental plasticity. The archaeological and paleontological records show gray wolf continuous presence for at least the last 300,000 years. This continuous presence contrasts with genomic analysis, which suggest that all modern wolves and dogs descend from a common ancestral wolf population that existed as recently as 20,000 years ago. These analyses indicate a population bottleneck, followed by a rapid radiation from an ancestral population at a time during, or just after, the last glacial maximum. This implies that the original wolf populations were outcompeted by a new type of wolf which replaced them. However, the geographic origin of this radiation is not known. Fossil record. The fossil record for ancient vertebrates is composed of rarely occurring fragments from which it is often impossible to obtain genetic material. Researchers are limited to morphologic analysis, but it is difficult to estimate the intraspecies and interspecies variations and relationships that existed between specimens across time and place. Some observations are debated by researchers who do not always agree, and hypotheses that are supported by some authors are challenged by others. There is general agreement on the most ancient record, which shows that feliforms and caniforms emerged within the superfamily Carnivoromorpha 43 million years before present, YBP. The caniforms included the fox-like genus Leptosian, whose various species existed from 34 million YBP before branching 11.9 million YBP into vulpes, foxes, and canini, canines. The jackal-sized Usean existed in North America from 10 million YBP, and by the early Pliocene about 6 to 5 million YBP the coyote-like Usean divisi invaded Eurasia. In North America it gave rise to early Canis, which first appeared in the Miocene, 6 million YBP, in southwestern US and Mexico. By 5 million YBP the larger Canis lepophagus appeared in the same region. P58. The canids that had immigrated from North America to Eurasia, Usean, vulpes, and nycteroutes, were small to medium-sized predators during the late Miocene and early Pliocene, but they were not the top predators. The position of the canids would change with the arrival of Canis to become a dominant predator across the Holarctic. The wolf-sized C. chilensis appeared in northern China in the mid-Pliocene around 4 to 3 million YBP. P148. The large wolf-sized Canis appeared in the Middle Pliocene about 3 million years ago in the Yushe Basin, Shanxi Province, China. By 2.5 million years ago its range included the Nihuen Basin in Yangyuan County, Hebei, China, and Kuruksay, Tajikistan. This was followed by an explosion of Canis evolution across Eurasia in the early Pleistocene around 1.8 million YBP and what is commonly referred to as the wolf event. It is associated with the formation of the Mammoth Steppe and continental glaciation. Canis spread to Europe in the forms of C. arnensis, C. etruscus, and C. falconeri. P148. The fossil record is incomplete, but it is likely that wolves arose from a population of small, early canids. P241. Morphological evidence P239 and genetic evidence both suggest that wolves evolved during the Pliocene and early Pleistocene eras from the same lineage that also led to the coyote. P239 with fossil specimens indicating that the coyote and the wolf diverged from a common ancestor 1.5 million years ago. P240 The ancestor of the jackal and the other extant members of the genus Canis had split from the lineage before this time. P240. After this separation from a common ancestor, the species that were believed to be involved in the further evolution of the wolf and coyote, and the beliefs of some paleontologists, diverged. P240 A number of researchers believe that the lines of C. priscolaterans, C. etruscus, C. rufus and C. lupus were components involved in some way that lead to the modern wolf and coyote. P240. Canis lepophagus. Canis lepophagus lived in the early Pliocene in North America. Curtin proposed that the Blankensia lepophagus derived from smaller Miocene Canis species in North America. It then became widespread across Eurasia, where it was either identical to, or closely related with, C. arnensis of Europe. P241. Johnston describes C. lepophagus as having a more slender skull and skeleton than in the modern coyote. 385 Robert M. Novak found that the early populations had small, delicate and narrowly proportioned skulls that resemble small coyotes and appear to be ancestral to coelatrans. P241 Johnson noted that some specimens found in Cedar Canyon, Texas had larger, broader skulls, and along with other fragments Novak suggested that these were evolving into wolves. P241 Tedford disagreed with previous authors and found that its cranio-dental morphology lacked some characteristics that are shared by C. lupus and coelatrans, and therefore there was not a close relationship, but it did suggest C. lepophagus was the ancestor of both wolves and coyotes. P119 Canis priscolatrans. Canis priscolatrans lived in the late Pliocene early Pleistocene in North America. The first definite wolf appeared in the late Blancan/early Irvingtonian, P240 and named C. priscolatrans that was either very close to or a synonym for Canis edwardii. P241:82 It resembled C. rufus in cranial size and proportions but with more complex dentition. P241 However, there are no fossils of C. rufus until the late Rancho-Labrian. P242 Curtin was uncertain if C. priscolatrans derived from C. lepophagus and C. arnensis, but believed that C. priscolatrans was a population of large coyotes that were ancestral to Rancho-Labrian and recent coelatrans. He noted that C. arnensis of Europe showed striking similarities to C. priscolatrans, and they could represent what once was a Holarctic population of coyotes. P27 Novak disagreed, and believed that C. priscolatrans was a counterpart to the European C. etruscus. Curtin later proposed that both C. priscolatrans and C. etruscus were part of a group which led to C. lupus, but was not sure if they evolved separately from C. lepophagus or a possible common ancestor that was derived from C. lepophagus. The remains of the larger coyote like Canis edwardii have been found in the later Pliocene in the southwestern USA along with C. lepophagus, which indicates a descent. P60 Tedford recognized C. edwardii and found that the cranio-dental morphology of C. priscolatrans fell inside that of C. edwardii such that the species name C. priscolatrans was doubtful. Nomen dubium, P131. Canis ambrosteri. The North American wolves became larger, with tooth specimens indicating that C. priscolatrans diverged into the large wolf C. ambrosteri. P242 during the Middle Pleistocene in North America. Robert A. Martin disagreed, and believed that C. ambrosteri was C. lupus. Novak disagreed with Martin and proposed that C. ambrosteri was not related to C. lupus but C. priscolatrans, which then gave rise to C. dirus. Tedford proposed that the South American C. gitzi and C. naringi shared dental and cranial similarities developed for hypercarnivory, suggesting C. ambrosteri was the common ancestor of C. gitzi, C. naringi and C. dirus. 148 Canis dirus. In 1908 the paleontologist John Campbell Merriam began retrieving numerous fossilized bone fragments of a large wolf from the Rancho La Brea tar pits. By 1912 he had found a skeleton sufficiently complete to be able to formally recognize these and the previously found specimens under the name C. dirus, Liddy 1858. Canis dirus lived in the late Pleistocene to early Holocene in North and South America and was the largest of all Canis species. 52 In 1987, new hypothesis proposed that a mammal population could give rise to a larger form called a hypermorph during times when food was abundant. But when food later became scarce the hypermorph would either adapt to a smaller form or go extinct. This hypothesis might explain the large body sizes found in many late Pleistocene mammals compared to their modern counterparts. Both extinction and speciation, a new species splitting from an older one, could occur together during periods of climatic extremes. Gloria de Goulet agreed with Martin and further proposed that this hypothesis might explain the sudden appearance of C. dirus in North America, and that because of the similarities in their skull shapes that C. lupus gave rise to the C. dirus hypermorph due to abundant game, a stable environment, and large competitors. Novak, Curtin and Berta disagreed with Goulet and proposed that C. dirus was not derived from C. lupus. The three noted paleontologists Xiaoming Wang, R. H. Tedford and R. M. Novak have all proposed that C. dirus had evolved from C. ambrosteri, P52:181 with Novak stating that there were specimens from Cumberland Cave, Maryland that indicated C. ambrosteri diverging into C. dirus. P243 The two taxa share a number of characteristics, synapomorphy, which suggests an origin of C. Dirus in the late Irvingtonian in the open terrain in the mid-continent, and then later expanding eastward and displacing its ancestor C. ambrosteri. 181. Dirus lupus hybrids. Merriam named 3 unusual species based on specimens recovered from the Rancho La Brea tar pits. They were regarded by Novak as taxonomic synonyms for Canis lupus. Canis occidentalis furlongi, Merriam 1910, was described as a wolf considerably smaller than the dire wolf and more closely related to the timber wolf Canis lupus occidentalis. However, its premolar P4, upper carnassials, were massive, and the hypocone of the molar M1 was larger than that of the dire wolf. One specimen's teeth and palate are described as being between the dire wolf and Canis lupus occidentalis. Novak proposed the name Canis lupus furlongi as he believed that it was a subspecies of the gray wolf. There is the possibility that wolves living in marginal areas led to dire wolf/gray wolf hybrids. Canis milleri, Merriam 1912, the Miller wolf, was as large as the timber wolf but with a shorter and heavier head. Its skull and dentition were described as being intermediate between Canis lupus occidentalis and the dire wolf. Its skull differed from occidentalis due to its wider skull, especially at the palate, and the size of its P4 and M1 were much larger than any known timber wolf, with the P4 approaching that of the dire wolf in size. It is regarded by Novak as a taxonomic synonym of Canis lupus furlongi. Enossian milleri, Merriam 1918, was described as a wolf different from the dire wolf by its smaller size, low sagittal crest, and a less prominent inion, but closer to the dire wolf than the timber wolf. Only one specimen was found. It is regarded by Novak as a taxonomic synonym of Canis lupus furlongi. Canis mosbachensis. Canis mosbachensis, sometimes known as the Mosbach wolf, is an extinct small wolf that once inhabited Eurasia from the Middle Pleistocene era to the Late Pleistocene. The phylogenetic descent of the extant wolf C. lupus from C. etruscus through C. mosbachensis is widely accepted. 239-245 In 2010, a study found that the diversity of the Canis group decreased by the end of the early Pleistocene to Middle Pleistocene and was limited in Eurasia to the small wolves of the C. mosbachensis C. variabilis group that were a comparable size to the extant Indian wolf, Canis lupus palipes, and the large hypercarnivorous Canis Xanasian, lichnoids that was comparable in size to extant northern gray wolves. Canis variabilis. In 2012, a study of the wolf-like Canis species of ancient China under the direction of Xiaoming Wang found that these were all quite close to C. lupus in both dental and postcranial dimensions except for Canis variabilis, which was very strange compared to other Canis in China as it had much smaller cranio-dental dimensions than earlier and later species. The study concluded that it is very likely that this species is the ancestor of the domestic dog Canis familiaris, a hypothesis that has been proposed by previous authors. Canis chiliensis. Wang and Tedford proposed that the genus Canis was the descendant of the coyote-like Yellowstone, and its remains first appeared in the Miocene, 6 million YBP, in southwestern USA and Mexico. By the Pliocene, 5 million YBP. The larger Canis lepophagus appeared in the same region, and by the early Pleistocene, 1 million YBP, Canis latrans, the coyote, was in existence. They proposed that the progression from Yellowstone to C. lepophagus to the coyote was linear evolution. P58 Additionally, C. edwardii, C. latrans and C. aureus formed together a small clade, and because C. edwardii appeared earliest spanning the mid-Blancan, late Pliocene, to the close of the Irvingtonian, late Pleistocene, it is proposed as the ancestor. P175, 180. Novak and Tedford also believed that it was possible for C. lupus to have been derived from a Miocene or Pliocene canid line that preceded and was separate from C. lepophagus. Based on morphology from China, the Pliocene wolf C. chiliensis may have been the ancestor for both C. armbrusteri and C. lupus before their migration into North America. P148, P181 C. chiliensis appears to be more primitive and smaller than C. lupus, and measurements of its skull and teeth are similar to C. lupus, but those of its postcranial elements are smaller. C. armbrusteri appeared in North America in the Middle Pleistocene and is a wolf-like form larger than any Canis at that time. At the end of the most recent glacial retreat during the past 30,000 years, warming melted the glacial barriers across northern Canada, allowing Arctic mammals to extend their range into mid-latitude North America, including elk, caribou, bison, and the gray wolf. P61. In Eurasia during the Middle Pleistocene, C. falconeri gave rise to the hypercarnivore genus Senecio, which then gave rise to genus Kuan, the dhole, and genus Lycaon, the African hunting dog. P105,149. Just before the appearance of C. dirus, North America was invaded by genus Senecio that was as large as C. dirus and more hypercarnivorous. The fossil record shows them as rare, and it is assumed that they could not compete with the newly derived C. dirus. P60. The large wolf C. antoni from late Pliocene to early Pleistocene China was assessed as being a variation within C. Chilensis, P197, and the large wolf C. falconeri occurred abruptly in Europe in the early Pleistocene, perhaps representing a westward extension of C. antoni. P181. Canis lupus. The earliest Canis lupus specimen was a fossil tooth discovered at Old Crow, Yukon, Canada. The specimen was found in sediment dated 1 million YBP, however the geological attribution of this sediment is questioned. Slightly younger specimens were discovered at Cripple Creek Sump, Fairbanks, Alaska, in strata dated 810,000 YBP. Both discoveries point to an origin of these wolves in East Thuringia during the Middle Pleistocene. In France, the subspecies C. L. lunellensis fauniferae, 1971, discovered at Lunel-Viel, Aero, dated 400 to 350,000 YBP, C. L. santinisiensis argent, 1991, from Santinay, Côte d'Or, dated to 200,000 YBP, and C. lupus maximus Budati Malene, 2012, from Joran's Cave, Nez Pools, Corres, dated 31,000 YBP, show a progressive increase in size and are proposed to be chronos subspecies. In Italy, the earliest Canis lupus specimens were found at La Poldrare di Sicanibio, 20 km northwest of Rome, in strata dated 340,000 to 320,000 YBP. In 2017, a study found that the dimensions of the upper and lower carnassial teeth of the early Holocene Italian wolf are close to those of C. L. maximus. Fluctuations in the size of C. lupus carnassial teeth correlate with the spread of megafauna. The Italian wolf underwent a reduction in body size with the loss of the red deer in Italy during the Renaissance. The proposed lineages: Canis cf. familiaris, Paleolithic dog. There are a number of recently discovered specimens which are proposed as being Paleolithic dogs, however their taxonomy is debated. These have been found in either Europe or Siberia and date 40,000 to 17,000 YBP. They include Hulafels in Germany, Gayet caves in Belgium, Predmosty in the Czech Republic, and 4 sites in Russia: Razboynica Cave, Kostyonkiate, Ulakansular, and Iliseyevichi 1. Paw prints from Chauvet Cave in France dated 26,000 YBP are suggested as being those of a dog, however these have been challenged as being left by a wolf. Paleolithic dogs were directly associated with human hunting camps in Europe over 30,000 YBP, and it is proposed that they were domesticated. They are also proposed to be either a proto-dog and the ancestor of the domestic dog, or a type of wolf unknown to science. Canis lupus familiaris, domestic dog. In 2002, a study was undertaken into the fossil skulls of 2 large canids that had been found buried within meters of the doorway of what was once a mammoth bone hut at the Iliseyevichi Upper Paleolithic site in the Bryansk region on the Russian plain, and using an accepted morphologically based definition of domestication declared them to be Ice Age dogs. The carbon dating gave a calendar year age estimate that ranged between 16,945 to 13,905 YBP. In 2013, a study looked at one of these skulls and its mitochondrial DNA sequence was identified as Canis lupus familiaris. In 2015, a zooarchaeologist stated that in terms of phenotypes, dogs and wolves are fundamentally different animals. In 1986, a study of skull morphology found that the domestic dog is morphologically distinct from all other canids except the wolf-like canids. The difference in size and proportion between some breeds are as great as those between any wild genera, but all dogs are clearly members of the same species. In 2010, a study of dog skull shape compared to extant carnivorans proposed that the greatest shape distances between dog breeds clearly surpass the maximum divergence between species in the carnivora. Moreover, domestic dogs occupy a range of novel shapes outside the domain of wild carnivorans. The domestic dog compared to the wolf shows the greatest variation in the size and shape of the skull, Evans 1979, that range from 7 to 28 centimeters in length, McGreevy 2004. Wolves are dolichocephalic, long-skulled, but not as extreme as some breeds of such as greyhounds and Russian wolfhounds, McGreevy 2004. Canine brachycephaly, Short-skulledness is found only in domestic dogs and is related to pedomorphosis. Goodwin 1997. Puppies are born with short snouts, with the longer skull of dolichocephalic dogs emerging in later development. Coppinger 1995. Other differences in head shape between brachycephalic and dolichocephalic dogs include changes in the craniofacial angle between the basilar axis and hard palate. Regatten 1993. Morphology of the temporomandibular joint. Dickey 2001, and radiographic anatomy of the cribriform plate. Schwartz 2000. Novak indicated that orbital angle of the eye socket is an important characteristic defining the difference between the dog and the wolf, with the wolf having the lower angle. Novak compared the orbital angles of 4 North American canines, including the Indian dog, and produced the following values in degrees: coyote 42.8, wolf 42.8, dog 52.9, dire wolf 53.1. The orbital angle of the eye socket was clearly larger in the dog than in the coyote and the wolf. Why it was almost the same as that of the dire wolf was not commented on. Many authors have concluded that compared to the adult extant wolf, the adult domestic dog has a relatively reduced rostrum, front part of the skull, an elevated frontal bone, a wider palate, a broader cranium, and smaller teeth. Hildebrand 1954, Cluttenbrock, Corbett and Hills 1976, Olson 1985, Wayne 1986, Hemmer 1990, Maury 1990. Other authors have disagreed and have stated that these traits can overlap and vary within the two. Crockford 1999, Harrison 1973. Wolf cubs have similar relative skull proportions as adult dogs, and this was proposed as evidence that the domestic dog is a neotenic wolf. This was proposed to be due to either human selection for juvenile appearance or due to a pleiotropic effect as a result of selection for juvenile behavior. Cluttenbrock 1977, Bayayev 1979, Wayne 1986, Coppinger and Schneider 1995. Wayne, 1986, concluded that his dog samples did not have significant relative shortening of the rostrum compared to wolves, calling this identification feature into question. A 2004 study that used 310 wolf skulls and over 700 dog skulls representing 100 breeds concluded that the evolution of dog skulls can generally not be described by heterochronic processes such as neoteny, although some pedomorphic dog breeds have skulls that resemble the skulls of juvenile wolves. Dogs are not pedomorphic wolves. Compared to the wolf, dog dentition is relatively less robust. Olson 1985, Hammer 1990, which is proposed to be due to the relaxation of natural selection when wolves became commensal scavengers, or to artificial selection. Olson 1985, Cluttenbrock 1995. However, Kieser and Gronewald, 1992, compared the mandibulodental measurements of jackals, Ciadestus, C. mesomelas, and Cape foxes, Vulpes chama, to equivalent-sized dogs and found that the canines of these other canids tended to be slightly smaller and their second molars larger compared to dogs. Otherwise the proportions were essentially the same in all species. They concluded that the teeth of canids appear to have evolved in concert with one another and relatively independently of differences in dimorphism, size or functional demands. This calls into question the assumption that dog teeth are relatively small due to recent selection, suggesting that dog dentition is plesiomorphic from an ancestor that was smaller than the wolf. The reduced body size of the early dog compared to a wolf is thought due to niche selection. Olson 1985, Maury 1992, Coppinger and Coppinger 2001. Maury, 1992-199, states that results are consistent with the hypothesis that early domestic dogs are evolutionary pedomorphs, products of strong selection for ontogenetically channeled size reduction and alterations of reproductive timing associated with a new domestic way of life. However, in a domestication experiment, the domesticated foxes remained the same size as unselected foxes. Trutt 1999-167. Wayne, 1986, concluded that the dog is closer in skull morphology to Coelatrans, C. aureus, C. adestus, C. mesomelas, Kuan alpinus and Lycaon pictus than to the wolf. Dar, 1942, concluded that the shape of the dog brain case is closer to that of the coyote than to that of the wolf. Manuel and Baker, 1983, reviewed Dar's work with the addition of dental data for canids and concluded that the dog ancestor was probably within the range of 13.6 to 20.5 kilograms, which is smaller than the range 27 to 54 kilograms for extant wolves, Mech 1970, and is comparable with the dingo. The auditory bulla of the dog is relatively smaller and flatter than that of the wolf. Harrison 1973, Cluttenbrock, Corbett and Hill 1976, Novak 1979, Olson 1985, Wayne 1986, which is proposed to be due to relaxed selection under domestication as the dog no longer required the acute hearing of the wolf. However, bulla's shape has been shown to facilitate increased sensitivity to specific frequencies, but shape and size may not be correlated with acuity. Ewer 1973. Therefore, the observed difference could be that the dog bulla has retained its ancestral shape. The ventral edge of the dog's horizontal ramus of the mandible has a convex curve that does not exist in the wolf. Olson 1985, Cluttenbrock 1995, and no discussion of this difference could be found in the literature. However, Byknevicius and Van Valkenburg, 1997, noticed that the horizontal ramus of bone-processing predators is thicker dorsoventrally at the point caudal to the site of bone processing. This thickening may have been a function for niche adaptation by the dog's ancestor. A description of the superficial brain morphology of jackals, C. mesomelas, C. aureus, coyotes, C. latrans, wolves, C. lupus, C. rufus, and dogs indicated that the cerebellum of the dog closely approximates that of the coyote, which is closely aligned with the jackals, and that the wolves show numerous brain traits distinct from the other species. Atkins and Dillon 1971. Wolves also have stereological and biochemical traits distinct from dogs. Leone and Weens 1956, Lauer, Count and Baker 1969. During the last glacial maximum, there was greater wolf genetic diversity than there is today, and within the Pleistocene gray wolf population the variations between local environments would have encouraged a range of wolf ecotypes that were genetically, morphologically and ecologically distinct from one another. One author has proposed that the most likely explanation for the different morphological characteristics of the dog compared to the wolf is that the dog's ancestor was adapted to a different niche than the wolf. Genetic record. DNA sequences. The mitochondria within each cell contain many copies of a small circular DNA genome, and in mammals it is 16,000 to 18,000 base pairs in length. A cell contains hundreds or thousands of mitochondria, and therefore the genes contained within those mitochondria are more abundant than the genes that occur in the nucleus of the cell. The abundance of mitochondrial DNA, MEDANA, is useful for the genetic analysis of ancient remains where the DNA has degraded. Mitochondrial DNA sequences have a higher mutation rate than the mutation rate of nuclear genes, and for mammals this rate is 5 to 10 times faster. The mitochondrial protein coding genes evolve much faster and are powerful markers for inferring evolution history at category levels such as families, genera, and species. However, they have evolved at a faster rate than other DNA markers, and there is a timing difference in its molecular clock that needs to be validated against other sources. The taxonomic status of uncertain species is better resolved through using nuclear DNA from the nucleus of the cell, which is more suitable for analyzing the recent history. In most cases, MEDANA is inherited from the maternal ancestor. Therefore, phylogenetic analysis of MEDANA sequences within species provides a history of maternal lineages that can be represented as a phylogenetic tree. The MEDANA sequences of the dog and wolf differ by only 0 to 12 substitutions within 261 base pairs, whereas dogs always differed from coyotes and jackals by at least 20 substitutions. This finding implies that the dog derived from the wolf and that there has been repeated back-crossing, or that the dog may have descended from a now extinct species of canid whose closest living relative is the modern wolf. Marker issue. Different DNA studies may give conflicting results because of the specimens selected, the technology used, and the assumptions made by the researchers. Anyone from a panel of genetic markers can be chosen for use in a study. The techniques used to extract, locate and compare genetic sequences can be applied using advances in technology, which allows researchers to observe longer lengths of base pairs that provide more data to give better phylogenetic resolution. Phylogenetic trees compiled using different genetic markers have given conflicting results on the relationship between the wolf, dog and coyote. One study based on SNPs, a single mutation, and another based on nuclear gene sequences, taken from the cell nucleus, showed dogs clustering with coyotes and separate from wolves. Another study based on SNPs showed wolves clustering with coyotes and separate from dogs. Other studies based on a number of markers show the more widely accepted result of wolves clustering with dogs separate from coyotes. These results demonstrate that caution is needed when interpreting the results provided by genetic markers. Timing issue. There are two key assumptions that are made for dating the divergence time per species: the generation time and the genetic mutation rate per generation. The time between generations for wolves is assumed to be 3 years based on the extant gray wolf, and 2 years for the dog based on the extant dog. One recent major study assumed a generation time of 2 years for the dog for as far back as 10,000 years ago, and then assumed a generation time of 3 years, the same as the wolf, before that to calculate a proposed divergence time between the two. In 2017, the wolf research scientist L. David Meck queried why evolutionary biologists were calculating the approximate time of the dog diverging from the wolf through using the wolf generation time of 3 years when published works using large data sets demonstrate a figure of 4.2 to 4.7 years. They were encouraged to recalculate their divergence dates accordingly. DNA studies are conducted but with the mutation rate as the dominant source of uncertainty. In 2005, Lindblad sequenced the first draft genome of the extant dog, and calculated a proposed mutation rate of 1 by 10⁻8 mutations per generation. In 2015, Skoglund was able to sequence the first draft genome of the 35,000 YBP Timur wolf and used its radiocarbon date to validate a proposed genetic mutation rate of 0.4 by 10⁻8 mutations per generation. The difference is a timing factor of 2.5. However, another study stated that because only one Pleistocene wolf specimen has so far been sequenced, then the result should be treated with caution, with that study then providing both estimates to calculate the proposed divergence times between the wolf and dog. However, in 2016 the mutation rate of the 4,800 YBP New-Grange dog matched that of the Timur wolf. Wolf-like canids. The wolf-like canids, the canid subfamily Caninae, are a group of large carnivores that are genetically closely related because their chromosomes number 78. The group includes genus Canis, Kuan, and Lycaon. The members of the dog: C. lupus familiaris, gray wolf; C. lupus, coyote; C. latrans, golden jackal; C. aureus, Ethiopian wolf; C. simensis, black-backed jackal; C. mesomelas, side-striped jackal; C. adestus, dole; Kuan alpinus, and African wild dog, Lycaon pictus. Newly proposed members include the red wolf, Canis rufus, eastern wolf, Canis lycaon, and African golden wolf, C. anthus. As they possess 78 chromosomes, all members of the genus Canis, coyotes, wolves, jackals, are chronologically indistinguishable from each other, and from the dole and the African hunting dog. P279 The members of Canis can potentially interbreed, and there is evidence that the Ethiopian wolf has hybridized with dogs. According to zoologist Reginald Pockett, a dole interbred with a golden jackal. The African hunting dog is large, highly mobile, known to disperse over large distances, and are rare throughout much of their geographical range, making opportunities for hybridization difficult. A study of the maternal mitochondrial DNA of the black-backed jackal could find no evidence of genotypes from the most likely mates, the side-striped jackal nor the golden jackal, indicating that male black-backed jackals had not bred with these. A search of the scientific literature could not find evidence of hybridization for the rare side-striped jackal. A DNA sequence alignment for the wolf-like canids gave a phylogenetic tree with the gray wolf and dog being the most closely related, followed by a close affiliation with the coyote, golden jackal and Ethiopian wolf, and the dog can hybridize in the wild with these three species. Next closest to this group are the dole and African wild dog that both have unique meat-slicing teeth, suggesting that this adaptation was later lost by the other members. The two African jackals are shown as the most basal members of this clade, which means that this tree is indicating an African origin for the clade. The tree illustrates the genotype-phenotype distinction, where a genotype is an organism's full hereditary information and a phenotype is an organism's actual observed properties, such as morphology, development, or behavior. By phenotype, the dole, genus Kuan, and the African hunting dog, genus Lycaon, are not classified as members of the genus Canis, but by genotype they are closer to dogs, wolves and coyotes than are the two genus Canis jackals, the side-striped jackal, C. adestus, and the black-backed jackal, C. mesomelas. In 2015, A study of mitochondrial genome sequences and nuclear genome sequences of African and Eurasian canids indicated that extant wolf-like canids had colonized Africa from Eurasia at least 5 times throughout the Pliocene and Pleistocene, which is consistent with fossil evidence suggesting that much of the African canid diversity resulted from the immigration of Eurasian ancestors, likely coincident with Pleo-Pleistocene climatic oscillations between arid and humid conditions. Admixture with an extinct unknown canid in 2018. Whole genome sequencing was used to compare members of genus Canis, along with the dhole, Cuon alpinus, and the African hunting dog, Lycaon pictus. There is evidence of gene flow between African golden wolves, golden jackals, and gray wolves. The study suggests that the African golden wolf is a descendant of a genetically admixed canid of 72% gray wolf and 28% Ethiopian wolf ancestry, and that the Ethiopian wolf once had a wider range in Africa. One African golden wolf from the Egyptian Sinai Peninsula showed high admixture with the Middle Eastern gray wolves and dogs, highlighting the role of the land bridge between the African and Eurasian continents in canid evolution. There is evidence of gene flow between golden jackals and Middle Eastern wolves, less so with European and Asian wolves, and least with North American wolves. The study proposes that the golden jackal ancestry found in North American wolves may have occurred before the divergence of the Eurasian and North American gray wolves. The study indicates that the common ancestor of the coyote and gray wolf has genetically admixed with a ghost population of an extinct unidentified canid. The canid is genetically close to the dhole and has evolved after the divergence of the African hunting dog from the other canid species. The basal position of the coyote compared to the wolf is proposed to be due to the coyote retaining more of the mitochondrial genome of this unknown canid. Similarly, a museum specimen of a wolf from southern China collected since 1963 showed a genome that was 12 to 14% admixed from this unknown canid. 2 wolf haplogroups. A haplotype, haploid genotype, is a group of genes in an organism that are inherited together from a single parent. A haplogroup is a group of similar haplotypes that share a common ancestor with a single nucleotide polymorphism, a mutation. Mitochondrial DNA passes along a maternal lineage that can date back thousands of years. In 2010, a study compared DNA sequences that were 230 base pairs in length from the mitochondrial control region of 24 ancient wolf specimens from Western Europe dated between 44,000 to 1,200 YBP with those of modern gray wolves. Most of the sequences could be represented on a phylogenetic tree. However, the haplotypes of the Himalayan wolf and the Indian gray wolf could not because they were 8 mutations apart from the other wolves, indicating distinct lineages which had previously been found in other studies. The study found that there were 75 different gray wolf Madonna haplotypes that include 23 in Europe, 30 in Asia, 18 in North America, 3 in both Europe and Asia, and 1 in both Europe and North America. These haplotypes could be allocated into 2 haplogroups that were separated from each other by 5 mutations. Haplogroup 1 formed a monophyletic clade, indicating that they all carried the same mutation inherited from a single female ancestor. All other haplotypes were basal in the tree, and these formed 2 to 3 smaller clades that were assigned to haplogroup 2 that was not monophyletic. Haplogroups 1 and 2 could be found spread across Eurasia, but only haplogroup 1 could be found in North America. The ancient wolf samples from Western Europe differed from modern wolves by 1 to 10 mutations, and all belonged to haplogroup 2, indicating a haplogroup 2 predominance in this region for over 40,000 years before and after the last glacial maximum. Data comparison of current and past frequencies indicated that in Europe haplogroup 2 became outnumbered by haplogroup 1 over the past several thousand years, but in North America haplogroup 2 became extinct and was replaced by haplogroup 1 after the last glacial maximum. Access into North America was available between 20,000 to 11,000 years ago after the Wisconsin glaciation had retreated but before the Bering Land Bridge became inundated by the sea. Therefore, haplogroup 1 was able to enter into North America during this period. Stable isotope analysis conducted on the bone of a specimen allows researchers to form conclusions about the diet, and therefore the ecology, of extinct wolf populations. This analysis suggests that the Pleistocene wolves from haplogroup 2 found in Thuringia and Belgium preyed mainly on Pleistocene megafauna, which became rare at the beginning of the Holocene 12,000 years ago. One study found the Thuringian wolf to be basal to all other gray wolves except for the extant Indian gray wolf and the extant Himalayan wolf. The Pleistocene Eurasian wolves have been found to be morphologically and genetically comparable to the Pleistocene Eastern Thuringian wolves, with some of the ancient European and Thuringian wolves sharing a common haplotype, A17, which makes ecological similarity likely. Two ancient wolves from the Ukraine dated around 30,000 YBP and the 33,000 YBP Altai dog had the same sequence as 6 Burundian wolves, and another from the Czech Republic dated 44,000 YBP had the same sequence as 2 Burundian wolves. It has been proposed that the Pleistocene wolves across northern Eurasia and northern North America represented a continuous and almost panmictic population that was genetically and probably also ecologically distinct from the wolves living in this area today. The specialized Pleistocene wolves did not contribute to the genetic diversity of modern wolves, and the modern wolf populations across the Holarctic are likely to be the descendants of wolves from populations that came from more southern refuges. Extant haplogroup 2 wolves can be found in Italy, the Balkans and the Carpathian Mountains but rare elsewhere in Europe. In Asia, only 4 haplotypes have been identified as belonging to this haplogroup, and 2 of them occur in the Middle East. Haplogroup 2 did not become extinct in Europe, and if before the last glacial maximum haplogroup 2 was exclusively associated with the wolf ecomorph specialized in preying on megafauna, it would mean that in Europe it was capable of adapting to changing prey. In 2013, a mitochondrial DNA sequencing of ancient wolf-like canids revealed another separate lineage of 3 haplotypes, forming a haplogroup that was found in 3 late Pleistocene specimens from Belgium. However, it has not been detected in extant wolves. One of these was the Jade dog. Dissenting view. In 2016, a study was undertaken due to concerns that previous Madonna studies may have been conducted with insufficient genetic resolution or limited geographical coverage and had not included sufficient specimens from Russia, China, and the Middle East. The study compared a 582 base pair sequence of the mitochondrial control region, which gave twice the phylogenetic resolution of the 2010 study. The study compared the sequences of both modern wolves and ancient wolf specimens, including specimens from the remote areas of North America, Russia, and China. The study included the Timur wolves, the Jade dog, the Altai dog, Burundian wolves, and other ancient specimens. The study found 114 different wolf haplotypes among 314 sequences, with the new haplotypes being found in Siberia and China. The phylogenetic tree resolved into 19 clades that included both modern and ancient wolves, which showed that the most basal clades included the Indian grey wolf and the Himalayan wolf, with a subclade of wolves from China and Mongolia falling within the Himalayan wolf clade. The 2 most basal North American haplotypes included the Mexican wolf and the Vancouver Island wolf. However, the Vancouver Island wolf showed the same haplotype as a dog, which indicates admixture, with the dog lineage basal to all extant North American subspecies. In Europe, the 2 most genetically distinct haplotypes form the Iberian wolf and separately the Italian wolf that was positioned close to the ancient wolves. The Greenland wolves all belong to one haplotype that had been previously found among North American wolves and which indicates their origin from North America. The eastern wolf was confirmed as a coyote/wolf hybrid. Wolves found in the regions of the Chukotka Peninsula, the North Korean border, Amur Oblast in Caucasia showed the greatest genetic diversity and with close links to all other wolves found across the Holarctic. One ancient haplotype that had been found in Alaska, eastern Beringia 28,000 YBP, and Russia, Medvedya Bear Cave, Pechora area, northern Urals 18,000 YBP, was shared with some modern wolves found in China and Mongolia. The previous finding of 2 wolf haplogroups was not clearly delineated in this study, but it agreed that the genetic diversity of past wolves has been lost at the beginning of the Holocene in Alaska, Siberia, and Europe with limited overlap with modern wolves. For the ancient wolves of North America, instead of an extinction/replacement model suggested by a previous study, this study found substantial evidence of a population bottleneck in North America in which the ancient wolf diversity was almost lost around the beginning of the Holocene. No further elaboration in the study. In Eurasia, the loss of ancient lineages could not be simply explained and appears to have been slow across time, with the reasons unclear. Into America and Japan. In 2016, a study built on the work of another major study and analyzed the sequences of 12 genes that are located on the heavy strand of the mitochondrial genome of extinct and modern C. lupus. The study excluded the sequences of the divergent Himalayan wolf and the Indian grey wolf. The ancient specimens were radiocarbon dated and stratigraphically dated, and together with the sequences generated a time-based phylogenetic tree. From the tree, the study was able to infer the most recent common ancestor for all other C. lupus specimens, modern and extinct, was 80,000 YBP, and this date concurred with the earlier study. The study could find no evidence of a population bottleneck for wolves until a few thousand years ago. The phylogenetic tree showed the polyphily of American wolves. The Mexican wolf was divergent from other North American wolves, and these other North American wolves formed two closely related clades. A scenario consistent with the phylogenetic, ice sheet and sea level data was that during the Ice Age, when sea levels were at their lowest, there was a single wave of wolf colonization into North America, starting with the opening of the Bering Land Bridge 70,000 YBP and closing during the late glacial maximum of the Yukon corridor that ran through the division between the Laurentide ice sheet and the Cordilleran ice sheet 23,000 YBP. Mexican wolves were part of the single wave and either diverged from the other wolves before entering North America or once in North America due to the change in its environment. As wolves had been in the fossil record of North America, but modern wolves could trace their ancestry back only 80,000 years, the wolf haplotypes that were already in North America were replaced by these invaders, either through competitive displacement or through admixture. The replacement in North America of a basal population of wolves by a more recent one supported the findings of earlier studies. There possibly existed a panmictic wolf population with gene flow spanning Eurasia and North America until the closing of the ice sheets. Once the sheets closed, the southern wolves were isolated, and north of the sheets only the Beringian wolf existed. The land bridge became inundated by the sea 10,000 YBP, the sheets receded 12,000 to 6,000 YBP, the Beringian wolf went extinct and the southern wolves expanded to recolonize the rest of North America. All North American wolves are descended from those that were once isolated south of the ice sheets. However, much of their diversity was later lost during the 20th century. Studies using mitochondrial DNA have indicated that the wolves of coastal southeast Alaska are genetically distinct from inland gray wolves, reflecting a pattern also observed in other taxa. They show a phylogenetic relationship with extirpated wolves from the south, Oklahoma, indicating that these wolves are the last remains of a once widespread group that has been largely extirpated during the last century, and that the wolves of northern North America had originally expanded from southern refuges below the Wisconsin glaciation after the ice had melted at the end of the last glacial maximum. A whole genome DNA study indicated that all North American wolves were monophyletic and therefore are the descendants of a common ancestor. During the same period, the Soya Strait between Hokkaido and Sakhalin Island was dry for 75,000 years, and it was proposed that the extinct Ezo wolf, C. L. hattai, arrived on Hokkaido from Sakhalin. However, the sequences indicated that it arrived in Hokkaido less than 10,000 YBP. The Ezo wolf was closely related to one of the North American clades, but different to the more southerly Japanese wolf, C. L. hadophylax, that was basal to modern wolves. The Japanese wolf inhabited Kyushu, Shikoku, and Honshu Islands but not Hokkaido Island. This indicates that its ancestor may have migrated from the Asian continent through the Korean peninsula into Japan. The past sea levels of the Korean Strait together with the timing of the Japanese wolf sequences indicated that it arrived to the southern islands less than 20,000 YBP. The dog was a very successful invader of North America and had established a widespread ecological niche by the early Middle Holocene. There was no overlap in niche between the dog and the wolf in comparison to the dog and other North American canids. By the late Holocene, the dog's niche area was less in size than researchers had expected to find, indicating that it was limited by biotic factors. These regions include the northeast and northwest of the United States that correlate with the greatest densities of early human occupation, indicating that the dog had defected from the wolf niche to the human niche and explains why the dog's niche area was not as large as expected. The separation between dog and wolf may reflect the rapid rate in which domestication occurred, including the possibility of a second domestication event occurring in North America. Packs of wolves and hunter-gatherers hunt similar prey in a similar way within a similar group social structure that may have facilitated wolf domestication. The wolf was exterminated in the southern part of their historic geographical range in North America by the middle of the 20th century. An Madonna study of 34 wolf remains from North America dated between 1856 and 1915 found their genetic diversity to be twice that of modern wolves in these regions, and 2/3 of the haplotypes identified were unique. These results indicate that a historic population of several 100,000 wolves once existed in Mexico and the western U.S. Divergence with the coyote. In 1993, a study proposed that the wolves of North America display skull traits more similar towards the coyote than those wolves from Eurasia. In 2016, a whole genome DNA study proposed, based on the assumptions made, that all of the North American wolves and coyotes diverged from a common ancestor less than 6,000 to 117,000 years ago. The study also indicated that all North America wolves have a significant amount of coyote ancestry and all coyotes some degree of wolf ancestry, and that the red wolf and eastern wolf are highly admixed with different proportions of gray wolf and coyote ancestry. One test indicated a wolf/coyote divergence time of 51,000 years before present that matched other studies indicating that the extant wolf came into being around this time. Another test indicated that the red wolf diverged from the coyote between 55,000 to 117,000 years before present and the Great Lakes region wolf 32,000 years before present. Other tests and modeling showed various divergence ranges and the conclusion was a range of less than 6,000 and 117,000 years before present. This finding conflicts with the fossil record that indicates a coyote-like specimen dated to 1 million years before present. Domestic dog. The domestic dog, Canis lupus familiaris, is the most widely abundant large carnivore. Over the past million years, numerous wolf-like forms existed but their turnover has been high, and modern wolves are not the lineal ancestors of dogs. Although research had suggested that dogs and wolves were genetically very close relatives, later phylogenetic analysis strongly supported the hypothesis that dogs and wolves are reciprocally monophilic taxa that form two sister clades. This suggests that none of the modern wolf populations are related to the wolves that were first domesticated and the wolf ancestor of dogs is therefore presumed extinct. Recent mitochondrial DNA analyses of ancient and modern gray wolf specimens supports a pattern of population reduction and turnover. An alternate proposal is that during the ecological upheavals of the late Pleistocene all of the remaining members of a dwindling lineage joined humans. In 2016, a study investigated for the first time the population subdivisions, demography, and the relationships of gray wolves based on their whole genome sequences. The study indicated that the dog was a divergent subspecies of the gray wolf and was derived from a now extinct ghost population of late Pleistocene wolves, and the dog and the dingo are not separate species. The genome-wide phylogenetic tree indicated a genetic divergence between New World and Old World wolves, which was then followed by a divergence between the dog and Old World wolves 27,000 YBP-29,000 YBP. The dog forms a sister taxon with Eurasian gray wolves but not North American wolves. The dog had considerable pre-ancestry after its divergence from the Old World wolves before it separated into distinct lineages that are nearly as distinct from one another as they are from wolves. The study suggested that previous datings based on the divergence between wolves and coyotes of 1 million years ago using fossils of what appear to be coyote-like specimens may not reflect the ancestry of the modern forms. The study indicated that the Mexican wolf was also a divergent form of gray wolf, suggesting that may have been part of an early invasion into North America. The Tibetan wolf was found to be the most highly divergent of the Old World wolves, had suffered a historical population bottleneck and had only recently recolonized the Tibetan plateau. Glaciation may have caused its habitat loss, genetic isolation and local adaptation. The study indicated that there has been extensive genetic admixture between domestic dogs and wolves, with up to 25% of the genome of Old World wolves showing signs of dog ancestry, possibly as the result of gene flow from dogs into wolves that were ancestral to all modern wolves. There was evidence of significant gene flow between the European wolves plus the Israeli wolf with a Pocentian boxer, which suggests admixture between the lineages ancestral to these breeds and wolf populations. For the lowland Asian wolves, the Central Russian and East Russian wolves and all of the lowland Chinese wolves had significant gene flow with the Chinese indigenous dogs, the Tibetan Mastiff and the dingo. For the Highland Asian wolves, the Tibetan wolves did not show significant admixture with dogs, however, the Qinghai wolves had gene flow with the dingo and one of them had gene flow with the Chinese dogs. The New World wolves did not show any gene flow with the boxer, dingo or Chinese indigenous dogs but there was indication of gene flow between the Mexican wolf and the African Basenji. All species within the genus Canis, the wolf-like canids, are phylogenetically closely related with 78 chromosomes and can potentially interbreed. There was indication of gene flow into the golden jackal from the population ancestral to all wolves and dogs, 11.3% to 13.6%, and much lower rates, up to 2.8%, from extant wolf populations. The data indicated that all wolves shared similar population trajectories, followed by population decline that coincided with the expansion of modern humans worldwide and their technology for capturing large game. Late Pleistocene carnivores would have been social living in large prides, clans and packs in order to hunt the larger game available at that time, and these larger groups would have been more conspicuous targets for human persecutors. Large dogs accompanying the humans may have accelerated the rate of decline of carnivores that competed for game, therefore humans expanded across Eurasia, encountered wolves, domesticated some and possibly caused the decline of others. The study concluded that admixture had confounded the ability to make inferences about the place of dog domestication. Past studies based on SNPs, genome-wide similarities with Chinese wolves, and lower linkage disequilibrium might reflect regional admixture between dogs with wolves and gene flow between dog populations, with divergent dog breeds possibly maintaining more wolf ancestry in their genome. The study proposed that analysis of ancient DNA might be a better approach. In the same year, a study found that there were only 11 fixed genes that showed variation between wolves and dogs. These genes are thought to affect tameness and emotional processing ability. Another study provided a listing of all of the gray wolf and dog Madonna haplotypes combined in the one phylogenetic tree. In 2018, a study compared the sequences of 61,000 single nucleotide polymorphisms, mutations, taken from across the genome of gray wolves. The study indicated that there exists individual wolves of dog/wolf ancestry in most of the wolf populations of Eurasia but less so in North America. The hybridization has been occurring across different time scales and was not a recent event. Low-level hybridization did not reduce the wolf distinctiveness. Dingo. The dingo, Canis lupus dingo, refers to the dog found in Australia. The dingo is a divergent subspecies of the grey wolf and is not a separate species, and is considered genetically to be a basal member of the domestic dog clade. The genetic evidence indicates that the dingo originated from East Asian domestic dogs and was introduced through the Southeast Asian archipelago into Australia, with a common ancestry between the Australian dingo and the New Guinea singing dog. Timor wolf. In May 2015, a study was conducted on a partial rib bone of a wolf specimen, named Timor 1, found near the Bolshaya-Balaknaya River in the Timor Peninsula of Arctic North Asia, that was AMS radiocarbon dated to 34,900 YBP. The sample provided the first draft genome of the cell nucleus for a Pleistocene carnivore, and the sequence was identified as belonging to Canis lupus. Using the Timor 1 specimen's radiocarbon date, its genome sequence and that of a modern wolf, a direct estimate of the genome-wide mutation rate in dogs, wolves could be made to calculate the time of divergence. The data indicated that the previously unknown Timor 1 lineage was a wolf population separate to modern wolves and dogs and indicated that the Timor 1 genotype, gray wolves and dogs, diverged from a now extinct common ancestor before the peak of the last glacial maximum, 27,000 to 40,000 years ago. The separation of the dog and wolf did not have to coincide with selective breeding by humans. Such an early divergence is consistent with several paleontological reports of dog-like canines dated up to 36,000 YBP, as well as evidence that domesticated dogs most likely accompanied early colonizers into the Americas. Comparison to the gray wolf lineage indicated that Timor 1 was basal to gray wolves from the Middle East, China, Europe and North America but shared a substantial amount of history with the present-day gray wolves after their divergence from the coyote. This implies that the ancestry of the majority of gray wolf populations today stems from an ancestral population that lived less than 35,000 years ago but before the inundation of the Bering Land Bridge with the subsequent isolation of Eurasian and North American wolves. A comparison of the ancestry of the Timor 1 lineage to the dog lineage indicated that some modern dog breeds have a closer association with either the gray wolf or Timor 1 due to admixture. The Charlos wolfdog showed more association with the gray wolf, which is in agreement with the documented historical crossbreeding with gray wolves in this breed. Timor 1 shared more alleles, gene expressions, with those breeds that are associated with high latitudes, the Siberian Husky and Greenland dog that are also associated with Arctic human populations, and to a lesser extent the Shar-Pei and Finnish Spitz. An admixture graph of the Greenland dog indicates a best fit of 3.5% shared material, although an ancestry proportion ranging between 1.4% and 27.3% is consistent with the data. This indicates admixture between the Timor 1 population and the ancestral dog population of these four high-latitude breeds. These results can be explained either by a very early presence of dogs in northern Eurasia or by the genetic legacy of Timor 1 being preserved in northern wolf populations until the arrival of dogs at high latitudes. This introgression could have provided early dogs living in high latitudes with phenotypic variation beneficial for adaptation to a new and challenging environment. It also indicates that the ancestry of present-day dog breeds descends from more than one region. An attempt to explore admixture between Timor I and gray wolves produced unreliable results. As the Timor wolf had contributed to the genetic makeup of the Arctic breeds, a later study suggested that descendants of the Timor wolf survived until dogs were domesticated in Europe and arrived at high latitudes where they mixed with local wolves, and these both contributed to the modern Arctic breeds. Based on the most widely accepted oldest zooarchaeological dog remains, domestic dogs most likely arrived at high latitudes within the last 15,000 years. The mutation rates calibrated from both the Timor wolf and the New Grange dog genomes suggest that modern wolf and dog populations diverged from a common ancestor between 20,000 to 60,000 YBP. This indicates that either dogs were domesticated much earlier than their first appearance in the archaeological record, or they arrived in the Arctic early, or both. The finding of a second wolf specimen from the same area, Timor II, and dated to 42,000 YBP has also been reported but yielded only mitochondrial DNA. Canis variabilis. In 2015, a study looked at the mitochondrial control region sequences of 13 ancient canid remains and one modern wolf from 5 sites across Arctic northeast Siberia. The 14 canids revealed 9 mitochondrial haplotypes, 3 of which were on record and the others not reported before. The phylogenetic tree generated from the sequences showed that 4 of the Siberian canids dated 28,000 YBP and 1 Canis c.f. variabilis dated 360,000 YBP were highly divergent. The haplotype designated as S805, 28,000 YBP, from the Yana River was one mutation away from another haplotype S902, 8,000 YBP, that represents clade of the modern wolf and domestic dog lineages. Closely related to this haplotype was one that was found in the recently extinct Japanese wolf. Several ancient haplotypes were oriented around S805, including Canis cf variabilis, 360,000 YBP, Belgium, 36,000 YBP. The Gaye dog, Belgium, 30,000 YBP, and Konsteki, Russia, 22,000 YBP. Given the position of the S805 haplotype on the phylogenetic tree, it may potentially represent a direct link from the progenitor, including Canis cf variabilis, to the domestic dog and modern wolf lineages. The gray wolf is thought to be ancestral to the domestic dog, however its relationship to C. variabilis, and the genetic contribution of C. variabilis to the dog, is the subject of debate. The Jokov Island, 8,700 YBP, and Akim, 1,700 YBP, canid haplotypes fell within the domestic dog clade cluster with S805, and also share their haplotypes with, or are one mutation away from, the Tibetan wolf, C.L. Filchneri, and the recently extinct Japanese wolf, C.L. Hodophylax. This may indicate that these canids retain the genetic signature of admixture with regional wolf populations. Another haplotype designated as S504, 47,000 YBP, from Duvany Yar appeared on the phylogenetic tree as not being connected to wolves, both ancient and modern, yet ancestral to dogs, and may represent a genetic source for regional dogs. The authors concluded that the structure of the modern dog gene pool was contributed to from ancient Siberian wolves and possibly from Canis cf variabilis. Rise to dominant predator. In 2015, a study looked at the paleoecology of large carnivores across the Mammoth Steppe during the late Pleistocene by using stable isotope analysis of their fossil collagen to reconstruct their diets. Based on testing in Belgium, around 40,000 YBP the cave hyenas preyed on mammoth, woolly rhinoceros, horses and reindeer, with cave lions taking reindeer and young cave bears. Wolves appear to have been outcompeted by cave hyenas and had their diet restricted to chamois, giant deer and red deer. However, after the last glacial maximum around 14,000 YBP, wolves had access to all prey species. The cave lion was restricted to reindeer, and the cave hyena had gone extinct. The data suggests that the extinction of the cave hyena allowed the wolf to become the dominant predator rather than the cave lion, just before the cave lion's extinction. Another study indicated that the wolf thrived compared to the cave hyena when there was greater snow cover. Wolf population differences. The gray wolf Canis lupus is a highly adaptable species that is able to exist in a range of environments and which possesses a wide distribution across the Holarctic. Studies of modern gray wolves have identified distinct subpopulations that live in close proximity to each other. This variation in subpopulations is closely linked to differences in habitat, precipitation, temperature, vegetation, and prey specialization, which affect cranio-dental plasticity. The archaeological and paleontological records show their continuous presence for at least the last 300,000 years. This continuous wolf presence contrasts with genomic studies, which suggest that all modern wolves and dogs descend from a common ancestral wolf population that existed as recently as 20,000 years ago. These studies indicate that a population bottleneck was followed by a rapid radiation from an ancestral population at a time during, or just after, the last glacial maximum. This implies that the original wolf populations were outcompeted by a new type of wolf which replaced them. However, the geographic origin of this radiation is not known. Apart from domestication, humans have harmed the wolf by restricting its habitat through persecution. This has caused a dramatic decrease in its population size over the last 2 centuries. The shrinking of its habitats that overlap with those of close relatives such as dogs and coyotes have led to numerous occurrences of hybridization. These events, in addition to recent turnovers, extinctions and repopulations by other genotypes, has made the unraveling of the phylogeographic history of the wolf difficult. Ecotypes. An ecotype is a variant in which the phenotypic differences are too few or too subtle to warrant being classified as a subspecies. These can occur in the same geographic region where distinct habitats such as meadow, forest, swamp, and sand dunes provide ecological niches. Where similar ecological conditions occur in widely separated places, it is possible for a similar ecotype to occur. This is different to a subspecies, which may exist across a number of different habitats. In animals, ecotypes can be regarded as micro-subspecies that owe their differing characteristics to the effects of a very local environment. Ecotypes have no taxonomic rank. Gray wolves have a wide, natural distribution across the Holarctic that includes many different habitats, which can vary from the high Arctic to dense forests, open steppe and deserts. The genetic differences between different populations of gray wolves is tightly linked to the type of habitat in which they live. Differences in genetic markers among the Scandinavian wolf population has arisen in only just over a decade due to their small population size, which indicates that these differences are not dependent on a long time spent in isolation and that larger population patterns can evolve in just a few thousand years. These differences can also include fur color and density, and body size. The differences can also include behavior, as coastal wolves eat fish and tundra wolves migrate. These differences have been observed between 2 wolf populations that are living in close proximity. It has been shown that mountain wolves do not interbreed with nearby coastal wolves, and the Alps of France and Switzerland have been repopulated with wolves from the mountains of nearby Italy and from the faraway mountains of Croatia rather than from the nearer lowlands, which indicates that distance is not the driving force in differences between the 2 ecomorphs. In 2013, a genetic study found that the wolf population in Europe was divided along a north-south axis and formed 5 major clusters. 3 clusters were identified occupying southern and central Europe in Italy, the Carpathians, and the Dinaric Balkans. Another 2 clusters were identified occupying north-central Europe and the Ukrainian steppe. The Italian wolf consisted of an isolated population with low genetic diversity. Wolves from Croatia, Bulgaria, and Greece formed the Dinaric Balkans cluster. Wolves from Finland, Latvia, Belarus, Poland and Russia formed the North-Central Europe cluster with wolves from the Carpathians cluster, a mixture of wolves from the North-Central cluster and the Dinaric Balkans cluster. The wolves from the Carpathians were more similar to the wolves from the Ukrainian steppe than they were to wolves from North-Central Europe. These clusters may have been the result of expansion from glacial refugia, an adaptation to local environments, and landscape fragmentation and the killing of wolves in some areas by humans. In 2016, 2 studies compared the sequences of 42,000 single nucleotide polymorphisms in North American gray wolves and found that they formed 6 ecotypes. These 6 wolf ecotypes were named West Forest, Boreal Forest, Arctic, High Arctic, Baffin, and British Columbia. The studies found that precipitation and mean diurnal temperature range were the most influential variables on sequence variation. These findings were in accord with previous findings that precipitation influenced morphology, and that vegetation and habitat type influenced wolf differences. One of these studies found that the variation in 11 key genes affected wolf vision, sense of smell, hearing, coat color, metabolism, and immunity. The study identified 1,040 genes that are potentially under selection due to habitat variation, and therefore that there was evidence of local adaptation of the wolf ecotypes at a molecular level. Most notable was the positive selection of genes that influence vision, coat color, metabolism and immunity in the Arctic and high Arctic ecotypes, and that the British Columbia ecotype also has a unique set of adaptations. The local adaptation of a wolf ecotype most likely reflects a wolf's preference to remain in the type of habitat that it was born into. Ecological factors including habitat type, climate, prey specialization and predatory competition will greatly influence gray wolf genetic population structure and cranio-dental plasticity. During the last glacial maximum, there was greater wolf genetic diversity than there is today, and within the Pleistocene gray wolf population the variations between local environments would have encouraged a range of wolf ecotypes that were genetically, morphologically and ecologically distinct from one another. Pleistocene wolves. The oldest Canis remains found in Europe were from France and dated to 3.1 million YBP, followed by Canis cf. Etruscus, where cf. in Latin means confer, uncertain, from Italy dated to 2.2 million YBP. C. lupus first appeared in Italy during marine isotope stage 9, 337,000 YBP. In Britain, it was the only canid species present from Mies 7, 243,000 YBP, with the oldest record from Pont Newy Cave in North Wales. During the Ice Age, Britain was separated from Europe by only the Channel River. A study of Pleistocene C. lupus in Britain at different time periods found that its abilities to crush, slice meat and eat bone highlighted its cranio-dental plasticity. These responses to dietary changes showed species-wide dietary shifts, and not just local ecomorphs, in response to climatic and ecological variables. The survival of C. lupus during the Pleistocene can be attributed largely to its plastic cranio-dental morphology. During the last glacial maximum 20,000 YBP, the Pleistocene steppe stretched across northern and central Eurasia and through Beringia into North America. The Pleistocene wolves of Beringia, and perhaps those across the steppe, were adapted to this habitat. Their tooth and skull morphology indicates that they specialized in preying on now extinct Pleistocene megafauna, and their tooth wear indicates that their behavior was different to modern wolves. This highlights the success of C. lupus as a species in adapting to different environmental conditions. This gray wolf ecomorph became extinct at the end of the glaciation, along with the horse and other species on which it depended, and was replaced by wolves from southern North America. This indicates that specialized wolf ecomorphs can become extinct when their environment changes even though the habitat may still support other wolves. Wolves went through a population bottleneck 20,000 YBP that coincides with the last glacial maximum, which indicates that many wolf populations may have gone extinct at the same time as the Beringian wolves. There are a small number of Canis remains that have been found at Jade Cave, Belgium, 36,500 YBP, Razboynica Cave, Russia, 33,500 YBP, Kustenkiate, Russia, 33,500 to 26,500 YBP, Predmosty, Czech Republic, 31,000 YBP, and Elizayevich 1, Russia, 17,000 YBP. Based on cranial morphometric study of the characteristics thought to be associated with the domestication process, these have been proposed as early Paleolithic dogs. These characteristics of shortened rostrum, tooth crowding, and absence or rotation of premolars have been documented in both ancient and modern wolves. Rather than representing early dogs, these specimens may represent a morphologically distinct local, now extinct, population of wolves. Notes: extinct 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 population of wolves. Notes: extinct